Olá, gente, boa noite a todos. Primeiramente, desculpa pelo transtorno, que a gente acabou tendo um problema com a plataforma. Eu sou a Giovana, secretária da Liga Acadêmica de Pneumologia e Cirurgia Torácica da Unicid, e é com muito prazer que a gente inicia o segundo dia do nosso Congresso de Intoxicações Pulmonares. Lembrando que hoje também a gente vai disponibilizar as 3 palavras-chave, vocês vão precisar preencher o formulário para presença, e a gente vai divulgar essas palavras ao longo da palestra, tanto na tela quanto no chat.
Doutora Thalita, antes de você começar, a gente gostaria de fazer uma breve apresentação do seu currículo. Então, a doutora Thalita é fisioterapeuta na Actum Neuromodelação e Reabilitação, é professora do curso de graduação em Medicina da Unicid, é orientadora do programa de pós-graduação Medicina da Unifesp, pesquisadora do Grupo de Pesquisa e Aplicação Tecnológica e Reabilitação, pós-doutorado concluído em Ciências de Reabilitação, Oxford, segundo pós-doutorado em andamento do Programa de Ciências de Reabilitação da Faculdade de Medicina USP, doutorado sanduíche pela Unifesp e estágio no exterior na School of Public Health da Harvard University, em Boston, nos Estados Unidos. Visita técnica na Virgin University, Amsterdã, Holanda. Doutora Thalita, muito prazer em tê-la. Pode começar.
Muito obrigada. Eu que agradeço pelo convite, mais uma vez, para falar de um pouquinho desse tema. Fico bastante feliz por ter sido convidada. Eu já, por todos vocês, que eu respeito muito dessa liga, fico muito contente mesmo. Eu já quero deixar, assim, aberto para vocês, não é a minha principal área, então a gente pode conversar bastante durante. Se vocês tiverem perguntas, eu até deixei aberto aqui para eu ver o chat, para se vocês tiverem alguma pergunta, fiquem à vontade para fazer. A ideia, basicamente, aqui é falar mais sobre um experimento que eu fiz. Então, eu sou fisioterapeuta por formação, trabalho mais com a parte de neurofuncional, neurologia funcional, e aí eu fiz um doutorado sanduíche na School of Public Health da Harvard, e aí lá eu tive a oportunidade de trabalhar com as partículas secundárias e trabalhos com inflamação pulmonar, e aí eu vou mostrar mais para vocês o que foi essa vivência lá e os resultados que nós tivemos desses experimentos. Então vai ser um prazer falar com você e relembrar um pouco disso, do que foi esse meu doutorado sanduíche. Deixa eu abrir aqui a apresentação. Vocês conseguem me ouvir bem? Dá para ver bem? Vocês vão me falando, se der alguma...
A gente está te ouvindo e te vendo, tá tudo certo.
Então, tá bom. Bom, e aí tudo começou nesse experimento com a professora Viviane, que deu aula aqui ontem, então pode ser que tenha algumas coisas parecidas para quem assistiu. Então, a professora Viviane me abriu grandes portas nesse laboratório, onde eu fui fazer meu doutorado sanduíche, enquanto ela fazia o pós-doutorado dela. E aí eu fui no laboratório do Dr. John Godleski, eu não sei se dá para ver eu mexendo o mouse aqui, mas de qualquer forma, Dr. Godleski é esse senhorzinho de barba branca aí, e esse é todo o restante do pessoal que trabalhava com a gente, né, comigo, com a Viviane, alguns alunos de medicina, que faziam na época estágio, eles eram alunos de medicina da USP, faziam estágio lá, e o Dr. Alex, que está aqui na foto também, que vai falar para vocês mais tarde, né, ele fazia o pós-doutorado dele lá. Bom, para falar para vocês o que eu fiz, lá eu fui interessada em fazer esses experimentos, né, eu trabalho com variabilidade da frequência cardíaca, e que é uma análise de sistema nervoso autonômico, né, como que é essa modulação autonômica sobre o coração. E lá eu fiz basicamente isso também, que fazia parte do meu pós-doutorado, analisar a variabilidade da frequência cardíaca dos ratos expostos a poluentes, que eu vou explicar também para vocês o que a gente fazia. E aí, antes disso, né, claro que todo mundo tem o sonho de ir para fora, todo mundo tem um grande sonho de fazer parte de grandes centros, né, Mas eu, quando era pequena, meu maior sonho era ser a morena do Chan, cada um com seu sonho, né? E aí lá eu consegui realizar esse sonho, na Harvard Chan School of Public Health. Então, já que eu não consegui ser a morena do Chan, não tenho talento para isso, consegui fazer parte. E uma curiosidade para vocês. Quando eu fui lá para Harvard, ela se chamava Harvard School of Public Health, né, da Escola de Saúde Pública. E esse rapaz chamado T.H. Chan, ele foi um rapaz que doou 400 milhões de dólares para a Escola de Saúde Pública. Lá nos Estados Unidos, eles têm esse costume de, quando você se forma em uma instituição, se você tem muito sucesso, você doa dinheiro para a instituição, já que a Harvard não é pública, né, ela é uma universidade privada. Esse moço teve muito sucesso na vida e doou 400 milhões de dólares para Harvard na época que eu estava lá, e eles simplesmente mudaram o nome dessa escola, né, da Escola de Saúde Pública da Harvard para Harvard T.H. Chan School of Public Health, né. Então, espero que todos tenham sucesso. A gente muda o nome da Unicid para vocês, se vocês doarem 400 milhões de reais também. Então, fica aí a dica e eu desejo que vocês tenham tanto sucesso assim. Bom, e aí a Harvard fica em Boston. Também trouxe algumas fotos só para deixar vocês com bastante vontade e desejo de ir para lá. Então, Boston é uma cidade maravilhosa, que tem todas as estações do ano muito bem implementadas, né? Então, a primavera é bem florida, o verão é muito quente, apesar de a água não ser, né? A água é bem fria. O outono é bem clássico, né? Tem como diz a Sandy, as folhas caem no quintal. E o inverno é bem friozinho. Então, para quem quer ir, tem que aguentar bem o frio de lá. Então, para quem gosta de ver as estações, lá é bem, bem clássico. E ainda o que interessa. Então, qual que era a ideia dos experimentos de lá, né? Então, lá eles trabalhavam, um laboratório de ciências ambientais, né, de environmental health, de saúde ambiental. Então, no momento que eu estava lá, eles estavam trabalhando e tentando estudar justamente o efeito de material particulado, principalmente que fossem de veículos. Então, o que eles queriam avaliar? O efeito desses materiais particulados e dos gases de biocombustíveis no pulmão e no coração de ratos, né? Então, tendo em consideração que esses poluentes inalados, eles iam gerar uma inflamação pulmonar, consequentemente uma inflamação sistêmica, e isso traria resultados tanto no cérebro quanto no coração. Então, a gente ia achar, a hipótese é de que nós acharíamos esses efeitos autonômicos, né, uns defeitos, inclusive, autonômicos, que iam levar também a problemas no coração, tá? Então, a gente poderia ter o efeito direto desses componentes no coração, ou um efeito indireto, né, que pode acabar prejudicando o cérebro e, consequentemente, a modulação autonômica cardíaca. Então, nós avaliamos tanto a parte cardíaca, né, de pressão de ventrículo esquerdo, a parte de eletrocardiograma, quanto a gente avaliou também o sistema autonômico deles para ver o prejuízo que tinha. A gente avaliou também pulmão, e aí eu vou mostrar para vocês. Pensando nisso, que nós que moramos em áreas urbanas, principalmente, nós temos uma grande exposição a poluentes vindos de veículos, de biocombustíveis. Então, a ideia era saber desses poluentes de combustíveis. Esse é um experimento. O outro experimento era para saber, inclusive, se as diferentes misturas de combustíveis, se você tem etanol com uma porcentagem maior de gasolina, uma porcentagem menor de gasolina e etc., e o quanto isso afeta tanto o pulmão, tanto o sistema pulmonar, quanto o sistema cardíaco. Mas esse primeiro experimento, basicamente, era para avaliar sistema nervoso autônomo e coração, tá? Pensando nisso, que todo esse efeito metabólico ia levar a uma disfunções nos canais iônicos, uma disfunção elétrica no coração, um ritmo alterado, que pode levar a doenças cardíacas, um infarto e, consequentemente, a morte, tá? Isso está muito bem estabelecido. E aí a gente teve a ideia de... a gente não, mentira. Eles tiveram a ideia e eu fui fazer o experimento. Como que eles pegavam esses materiais particulados? Em Boston, tem uma dificuldade muito grande de achar ar poluído. Olha só que beleza. Então, a coleta do ar era feita dentro desses túneis que tinha lá, né? Então, tinha que procurar esses túneis para conseguir achar um ar relativamente poluído, para poder fazer os experimentos, né? Com material particulado de 2,5. Nós usamos ratos, né? Esses Progdowleys, rats, com um número de 6 em cada grupo, e fizemos 2 grupos. Um que era exposto somente a ar puro, e um outro grupo que era exposto a um material particulado com partículas secundárias, né? Foram feitas 8 semanas de exposição desses ratos, e eu vou mostrar para vocês tudo direitinho, para vocês terem uma ideia de como foi feito o experimento. Não, mentira, foram feitas 3 semanas, isso aqui foi uma outra questão de dieta lá, que foi feita, e uma monitoração por telemetria, tá? E aí, o que a gente fazia? Então, lá, a minha parte, basicamente, do experimento era de fazer essas cirurgias dos ratos. Então, a gente implantava tanto esse aparelho de telemetria, eu vou mostrar até alguns videozinhos para vocês, quem não tem muito estômago, eu peço para fechar os olhinhos daqui para frente. E aí eu fazia essas cirurgias de implantar os eletrodos para a gente pegar o eletrocardiograma e também o aparelho de telemetria que ia avaliar a pressão do ventrículo esquerdo deles, tá? Então a gente colocava aqui embaixo da pele alguns eletrodos, a gente passava por baixo ali da pelezinha dele, fazia a exposição também do diafragma, depois fazia a exposição do coração. E aí a gente colocava um catéter bem posicionado bem no ápice do coração. Então, aqui, o que a gente fazia? Eu abria aqui, a gente abria a pele, abria a barriguinha deles, aí aqui, quando a gente abre, tem exposto o diafragma, logo aqui embaixo, e a gente fazia o que eu fazia era uma incisão bem aqui no diafragma. Quando a gente abre o diafragma, a gente abria e expunha o coração, tá? Pulmãozinho batendo aqui. E aí a gente pegava bem no ápice mesmo aqui para colocar o catéter. Esse era o ponto que a gente colocava o catéter. Então eu vinha, segurava o coração, ancorava ele, botava um catéter ali bem para pegar o ventrículo esquerdo, tá? Esse era o catéter que a gente colocava, tá? Então ele vem aqui, a gente coloca, faz uma sutura, amarra o catéter mesmo ali na parede do coração e depois fechava tudo e depois fazia as análises. Então, eu vou mostrar para vocês. Esse era o aparelho de telemetria. Então, o que ele faz? A gente deixa ele dentro do rato, essa parte grandona aqui, e fica dentro do abdômen. E depois, quando a gente fecha o ratinho, tudo bonitinho, ele tem a captação por telemetria dos dados de pressão e do eletrocardiograma, que eu também vou mostrar para vocês. Então, aqui está o dispositivo. Então, a gente colocava ali no abdômen mesmo, bem embaixo do músculo e em cima do intestino. E aí, depois disso, que a gente fazia as incisões para colocar cada eletrodo, né, um de cada lado, nas posições corretas, para fazer o eletrocardiograma. Então, ali a gente fazia um buraquinho para poder passar ali, vou passar essa parte, que é só para ancorar o aparelho ali no músculo. Deixa eu ver se tem. Aqui a gente passava um tubo, passava através do músculo mesmo, para eu conseguir colocar o eletrodo de eletrocardiograma. Então, eu colocava o eletrodo ali, passava pelo músculo, e depois colocava ali subcutâneo, tá? Então, tinha que colocar ali embaixo depois da pele, embaixo da pele, para conseguir fazer a análise eletrocardiográfica depois. Aí vai um de cada lado, tá? Um do lado direito, o outro do lado esquerdo, para depois ter o dado certinho da eletrocardiografia. Essa daqui é a parte da incisão. Então, aqui a gente está olhando o diafragma, vai melhorar essa imagem, prometo para vocês. Então, aqui a gente faz a incisão no diafragma, com bastante cuidado para não pegar nada, nem um pedacinho do pulmão, nem um pedacinho do coração. E a gente tem que separar o diafragma para conseguir chegar ali no coração. Essas são partes bastante difíceis. O rato é anestesiado, tá? Passa por todas as questões éticas para ele não sofrer nem um pouquinho. Ele fica em ventilação mecânica, respirando por aparelhos, com anestesia o tempo todo, né, isoflorando. E então a gente tentava tirar o diafragma aí da frente para conseguir acessar o coração. Passar para frente. Aí aqui é o pulmão, batendo o pulmão, ventilando. E aqui a gente vai para a parte de acessar o coração. Então, o que a gente fazia? Eu tinha que ancorar o coração, então eu tinha que ir lá buscar o coração. E eu tinha que fazer um buraquinho exatamente onde eu tivesse acesso ao ventrículo esquerdo e não ao ventrículo direito, justamente porque a gente queria pegar a pressão do ventrículo esquerdo, né? Então, pesquei o coração, batendo rapidinho, graças a Deus. E aí eu tenho que realmente vir com uma agulha e fazer um furo nesse coração de um tamanho suficiente para eu conseguir colocar o catéter nele. Então, essa era toda a dificuldade. Tudo isso para a gente conseguir avaliar os efeitos da poluição depois dele. Deixa eu passar um pouquinho para frente. Deixa eu só pegar um momento do furinho. Aqui acho que só a parte do âncora. Tá. Então, desculpem, errei no vídeo. É esse aqui que, então eu venho com uma agulha, tá vendo? Pego ali no ápice do coração, faço um furinho. Só que nessa hora jorra muito sangue, porque a gente tá falando de ventrículo esquerdo, né? Então ele que é o responsável por levar o sangue ali para todo o corpo. Então eu tenho que pegar, ser bastante rápida, pegar o catéter e colocar ali onde eu fiz o furinho, tá? Inserir o catéter, esse catéter, a partir desse momento, faço os nozinhos, né, vou ancorar mesmo, numa sutura que eu fiz no coração, e a partir daí ele vai ficar lá avaliando a pressão do ventrículo esquerdo, tá? Então ali eu tô suturando esse catéter no coração, né, tô prendendo esse catéter no coração, tomar bastante cuidado para não rasgar nada. E a partir daí a gente sai fechando tudo. Então, ancorei o catéter aqui no coração, eu fico monitorando nesse momento, tem um painel, um monitor, e esse monitor é onde eu consigo ver se eu estou no ventrículo esquerdo ou não. Então, eu consigo ver exatamente se, pela curva do gráfico que eu vou mostrar para vocês, se eu atingir ventrículo esquerdo, se eu estou no ventrículo direito, se eu tenho que fazer uma nova incisão, se o catéter soltou. Então, eu consigo acompanhar tudo em tempo real. Depois a gente vem só fechando tudo. Então, eu venho, fecho o diafragma, deixo só um espacinho para o catéter, fecho também, solto aqui o externo deles, e aí, tá vendo? Eu venho só cortando tudo, depois eu saio fechando tudo isso, tá? Fecho o diafragma, fecho aqui o abdômen, fecho a pele, e aí a gente espera ele acordar, estuba, e vida que segue pro rato. E é isso que eu vejo. Então, aqui em cima eu tenho as curvas da pressão do ventrículo esquerdo. Se fosse um ventrículo direito, essa pressão ela é bem baixinha, ela é bem diferente. Então, quando a gente via que essa curva ficava baixinha porque a gente estava no ventrículo direito, tinha que voltar a furar de novo. E aqui a gente acompanha o eletrocardiograma, tá? Eu também vejo se o eletrocardiograma está bem posicionado, se foi tudo certinho. Aqui eu tenho os picos R's, né, de cada onda do eletrocardiograma, então a gente conseguia ver se estava tudo certo e podia deixar, continuar, fechar o rato e esperar ele acordar. Depois isso era o que a gente avaliava. Aqui, a partir desses picos de onda R, então, entre uma onda R e a outra, é que a gente fazia a análise do sistema nervoso autônomo. Então, pelo tempo em milissegundos entre uma onda e outra, a gente consegue saber se esse rato tem uma predominância simpática, parasimpática, dependendo de cada momento. Então, a gente tem batimentos ectópicos, né, que são uns diferentes dos outros por alguma coisa que aconteceu, e a gente consegue fazer toda a análise autonômica dele. Em repouso, antes e depois da exposição ao agente, né, ao material particulado, então a gente consegue fazer várias análises aqui com esse rato por telemetria. Então, só mostrando mais uma vez para vocês. Inclusive, a gente tinha vários outros, esse aparelho de telemetria, ele mostrava para a gente curvas de pressão, pressão por tempo, enfim, um monte de variável bem legal. E como que era o sistema de... Então, aqui tinha esses pletismógrafos, e esses pletismógrafos é o que faziam a coleta do dado. Então, o rato ficava ali em cima, aqui ele tá todo tímidozinho, dentro da casinha dele, e aí esse rato conseguia, ele tava com o aparelho já aqui dentro dele, e esse pletismógrafo é quem faz a captação dos dados de pressão, de variabilidade da frequência cardíaca, do eletrocardiograma, tudo isso, tá? Então, ele não precisa ficar preso a nenhum dispositivo. Aquele dispositivo que fica dentro dele passa todas essas variáveis. E eles ficavam aqui nesse sistema de exposição. Então, uma parte desses ratos ficava expostos ao ar, o ar que entra aqui, então ele só tem acesso ao ar que está aqui dentro dessa casinha. Esse ar, a metade dos ratos ficava expostos ao ar poluído, com material particulado, com materiais de combustíveis, e o outro grupo era exposto ao ar limpo, né? Então, esse aqui era o protocolo. Então, todos eles ficavam expostos só a ar puro, por 3 dias, e depois eles sofriam estresse, para justamente a gente ver como que o coração respondia. Qual que era o estresse? A gente colocava eles dentro de uma caixinha, uma cage deles mesmo, né? Uma caixinha da casinha deles, e a gente colocava um rato muito maior e agressor. E aí, a gente avaliava o estresse, a resposta dele ao estresse e a recuperação deles do estresse. E aí, depois, eles ficavam essas 4 semanas expostos o grupo que era do material particulado ficava exposto ao material particulado, ou quem era do grupo controle ficava expostos a ar puro, tá? E aí, no último dia, também expunha a estresse e depois fazia a necrópsia deles para pegar um pedacinho de cada parte do corpo. Aí é isso que a gente fazia. Então, tem um ratinho aqui em posição submissa, né? Quer dizer que ele tá com medo. Ele entrava nessa casinha. E o rato maior, que é o que é agressor aqui. Tinha uma outra também que a gente fazia, que era de colocar esses dispositivos aqui na nuca deles e esse catéter que ia no coração exatamente igual ao outro e fazia a injeção de nanopartículas aqui. Então, colocava as nanopartículas, elas iam lá para o ventrículo esquerdo deles também, para ver qual o caminho dessa nanopartícula, tá? E o último, que é a quimiluminescência. Resultado mesmo, eu só vou conseguir mostrar para vocês da quimiluminescência, porque os resultados desse primeiro estudo ainda não saíram, inclusive a culpa é do Alex, que vai falar para vocês aqui depois. É ótimo pôr a culpa em alguém. E da quimiluminescência já está pronto o resultado, eu vou mostrar para vocês. Então, esse aqui é um outro experimento. Essa aqui era a dark room, então a sala escura, que é uma sala que a gente avalia a espécie reativa de oxigênio, né, os ROS. E aqui eu fazia o experimento que eu vou mostrar para vocês, é bem mais simples, E só que durante a avaliação mesmo, quando a gente estava avaliando o rato, a gente tem que apagar todas as luzes dessa sala, ela realmente fica 100% escura, ela precisa ficar, né, porque eles captam luminóis. E inclusive a porta dela, a porta é uma porta giratória, assim, meio que baú da felicidade, assim, para justamente ninguém abrir e não entrar ar. Então é uma porta que girava, assim, bem escura mesmo. Não sinto saudade. E aí a gente fazia a análise. Nesse caso, o que a gente fazia? Fazia a instilação mesmo de material particulado. Isso aqui fazia antes desse procedimento aqui, tá? Isso aqui já é a parte da exposição. Esqueci, mas acho que a professora Viviane mostrou para vocês ontem também a questão da instilação. Fazia com um spray, fazia instilação direto na traqueia deles, para eles inalarem o ar poluído. O ar poluído desse experimento veio do Chile, e aí eu vou mostrar para vocês também. O que eles fizeram no Chile? Eles colocaram 2 carros, um carro brasileiro e um carro dos Estados Unidos, e eles colocaram também gasolinas diferentes nesses carros. E eles colocavam um filtro bem ali onde saía, no escapamento, para conseguir captar todos os poluentes que saíam direto do escapamento. E eu vou mostrar para vocês os diferentes tipos de gasolina. Então, com diferentes tipos de mistura de gasolina e etanol, depois eles mandavam esse material particulado para a gente, do Chile lá para Boston. A gente fazia toda a captação desses materiais particulados, misturava em um meio que imitava o surfactante do pulmão. Então, a gente fazia todo esse meio e misturava com material particulado nos ratos expostos. E nos ratos controle, a gente só fazia um meio que imitava o surfactante sem o material particulado. Esse material particulado a gente instilava com esse spray direto na traqueia deles. Uns ratos ficavam por 4 horas, outros ratos ficavam por 45 minutos depois da exposição. Ok, passou esse tempo de exposição, a gente simplesmente abria o ratinho, botava lá no ventilador mecânico com anestesia, e aí a gente colocava eles simplesmente expostos. Então aqui a gente vê o pulmãozinho, isso aqui é só um soro fisiológico, tá? Pulmãozinho respirando, coraçãozinho batendo, e aí eu colocava ou o pulmão exposto ou o coração. Então, aqui eu tenho o pulmão exposto, aqui eu tenho o coração exposto. Eu precisava fazer isso para colocar eles dentro da máquina, tá? Então, uma hora eu escondia outra hora eu escondia outro. Por quê? Eu queria saber, primeiro, qual que era a resposta no pulmão e depois qual que era a resposta no coração. A resposta com relação a estresse oxidativo, tá? Aí fazia essa exposição, uma hora deixando o pulmão, outra hora deixando o coração e colocava ele nessa máquina. Então aqui ele tá deitadinho, para quem não tá reconhecendo, aqui é a boca, aqui os bracinhos, e aqui tá o pulmão ou o coração exposto. A gente colocava dentro, ficava um buraquinho aqui em cima dessa máquina, e aí esse buraquinho é o que capta o estresse oxidativo, tá? Então assim que a gente expunha ou o coração ou o pulmão, para fazer a captação da quimiluminescência. E então surgiu esse artigo, que é a análise de partículas secundárias formadas pela exaustão dos veículos usando misturas de etanol e gasolina, para avaliar a produção de espécie reativa de oxigênio, tanto no pulmão quanto no coração, e avaliar a inflamação pulmonar. Tá? E aí, de novo, eu tinha esses tipos de gasolina, então eu tinha só gasolina, 0% de etanol, que foi chamada de E0, tinha gasolina com 10% de etanol, que é o E10, gasolina com 22% de etanol, que essas são geralmente as brasileiras, tá? E22, E100 e E10, se eu não me engano, e a gasolina com 85% de etanol. Uma outra diferença, a gente teve também, porque aqui tem 5, nós tivemos 6 grupos. Esses 6 grupos, por quê? 85% foi captada no verão e no inverno. Então, para ver a diferença dessa exaustão dos veículos no verão ou no inverno, se alguma produz mais ou menos inflamação pulmonar. Então, nós tivemos esses grupos e o grupo que era 100% etanol, tá? E para ver qual desse tipo de mistura, ou sem mistura nenhuma, né, só gasolina ou só etanol, produz mais ou menos inflamação pulmonar e produção de espécie reativa de oxigênio no coração e no pulmão. E aí, o que a gente encontrou? Aqui a gente viu que a gente teve a análise da quimiluminescência em si, né, no pulmão e no coração, e a análise do lavado broncoalveolar. Então, a gente viu a quantidade de macrófagos, neutrófilos, linfócitos e monócitos em ambos também, coração e pulmão, depois de 45 minutos e depois de 4 horas. Deixa eu mostrar isso aqui primeiro. Como conclusão, a gente encontrou que quando você tem só gasolina, quando você não tem nada de etanol, você tem um aumento da quantidade de estresse oxidativo no pulmão, assim como tem também quantidade maior de macrófagos e quantidade maior de linfócitos. A quantidade maior de macrófagos, neutrófilos e linfócitos foi achada também quando você tem 85% de etanol. Então, você tem gasolina com 85% de etanol. E também, quando você tem 100% de etanol, quando você não tem nada de gasolina, você tem um aumento maior no coração do estresse oxidativo. E geralmente após 45 minutos. Tá? Então, parece ser uma resposta muito rápida inflamatória, quando você tem exposição a materiais particulados com misturas de gasolina e etanol. Tá? Então, o que a gente achou como conclusão é que a gasolina sozinha e as misturas de etanol induzem o estresse oxidativo, tem uma maior geração de espécie reativa de oxigênio e inflamação pulmonar, que a gente achou por meio das células brancas, aqui macrófago, principalmente macrófago, neutrófilo e linfócito, no lavado broncoalveolar. Tá? E aí, o que a gente discute é justamente isso. A gente tem uma predominância maior de macrófagos, o que é esperado, né? Principalmente quando a gente pensa que eles vão ser responsáveis por remover as partículas, né, ali naquela região dos alvéolos, e vão também ativar outras células do sistema imune, como os neutrófilos, por exemplo, para migrar para o pulmão, para conseguir proteger de alguma forma, né? Então, a gente acha desde 45 minutos já respostas inflamatórias, né, e resposta de sistema imune quando você tem uma exposição a misturas de etanol e gasolina, tá? Bom, gente, basicamente acho que é isso, corri um pouquinho que a gente estava atrasada, então eu não sei se alguém tem alguma pergunta para fazer. Meninas?
Oi, Pro. Eu gostaria de agradecer a sua aula. Acho que foi muito boa, os vídeos foram muito didáticos, né? Ajudou a entender bastante. E é isso, foi muito boa a aula.
Muito obrigada.
Não consigo digitar para.
Bom, e agora nós vamos dar início aos sorteios, mas só lembrando antes que o nosso patrocinador Utilidades Clínicas disponibilizou um cupom de 15% de desconto que já foi colocado no chat. E agora a gente vai sortear 15 cupons de um mês da PebMed. Pode compartilhar, Gi. Então, o primeiro número foi o 212. vai conferir agora na planilha. R$212, Antonella Fracoli Rosseto. Próximo número, 88. Maria Eduarda Ninômia. Próximo número, 131. A Natália Aguiar, ela já ganhou, então a gente vai sortear mais um número depois. Próximo número, 153. Fernanda Mendonça de Paula Santos. Próximo número, 221. Andressa Silva de Oliveira. Próximo número, 139. A Vitória Espínola, ela já ganhou. Vamos sortear 2 números, então. Próximo número, 225. Letícia Seixas Mendes. Próximo número, 48. Ângelo Gutenberg Santos Lima. 109. A Thaís já ganhou, então a gente vai sortear 3. 203 agora. Próximo número. 203. Tatiane Viana Cavalcante. Próximo número, 137. Já foi sorteado, então serão sorteados mais 4. Próximo número, 194. Matheus França Cunha. Próximo número, 168. Jadson Pereira Dias. Próximo número, 93. Daiana Grosso Oliveira. Próximo número, 14. Edilene Dutra. Então, a gente vai sortear mais 4 agora. Mais 4 números. Primeiro número é o 39. O segundo número é o 50. O 39 foi o Pablo Rendi Fernandes. Próximo número, 50. Stephanie Kelly. Próximo número, 12. Bárbara 12. E o próximo é o 13, que é a Camila. Então, já foram os 15 sorteados da Whitebook e a gente vai agora dar início ao intervalo e voltar daqui 20 minutos. Então, às 8h20, a gente retorna com o nosso próximo palestrante, Dr. Alex.
Boa noite. Boa noite a todos. Então, retornamos para a última palestra do nosso evento. E, em nome de toda a comissão organizadora do Congresso Intoxicações Pulmonares, eu gostaria de agradecer ao Dr. Alex pelo material. Ok? O Dr. Alex, ele vai estar na nossa aula depois que a gente mostrar o vídeo. Dr. Alex é professor assistente da Universidade de Louisville, Kentucky. Tem pós-doutorado pela Universidade de Harvard em Saúde Pública e Fisiologia Integrada. PhD pela Universidade Carolina do Norte em Saúde Pública e Ciências de Saúde Ambiental. Mestrado em Ciências de Saúde Pública. Formado pela Faculdade Duke em Políticas de Ciências Ambientais. Eu gostaria de lembrá-los que o material que vamos expor contém dados ainda não publicados, e esses dados não podem ser compartilhados. Ficamos à disposição para as dúvidas nos comentários. A gente teve um problema com o som, só um minutinho. A aula do professor Alex é gravada, então, por isso, o material não pode ser compartilhado.
Hello from Louisville, Kentucky. My name is Alex Carl, and I'm an assistant professor in the Department of Physiology and at the Diabetes and Obesity Center at the University of Louisville School of Medicine. And I've done most of my recent research as part of the American Heart Association's Tobacco Regulation and Addiction Center. So today I'm going to talk about my research into e-cigarettes, and I titled it somewhat arbitrarily E-cig Discordia in honor of Discordia, the Roman goddess of strife, warfare, and chaos. And she's notorious for having started the Trojan War by creating a feud between Aphrodite and some other goddesses by throwing in a golden apple into a wedding. So at any rate, for some reason, Discordia struck me as relevant. At the center of her name is heart, actually, in Latin. And most of the research that I've been conducting has shown chaos in the heart induced by e-cigarettes. So that's what I'm going to focus on today, is sort of this introduction of chaos in the heart induced by e-cigarettes, primarily in mice here, but with a little bit of a translational approach as well at the end. So here in the United States, over the past, I guess, 5 to 10 years, we've seen a surge in e-cigarette use, and this has been especially evident among our youth, especially among high schoolers, but also middle schoolers. So high schoolers are generally about 14 to 18 years old in the United States, and they have reduced their use prevalence for combustible cigarettes, but the prevalence of e-cigarette use within the past 30 days has surged dramatically. And a lot of the recent surge is attributable to an e-cigarette called Juul, which you may be familiar with. And Juul did a very effective job of campaigning via advertisements for the attention of teenagers, as you can see here. So on the graph here, we've got data from the Centers for Disease Control in the United States showing the prevalence of e-cigarette use all the way up to a quarter of high schoolers using e-cigarettes. And this is concerning because we don't really know the cardiac impacts or the pulmonary impacts, in truth, of long-term e-cigarette use. But we also scarcely understand the acute effects of e-cigarette use on the heart and the lungs. So my research has delved into the cardiac impacts of e-cigarettes. But before I get into that, just a brief primer on e-cigarettes. There are 4 main types of e-cigarettes. The cig-alike was the first generation of e-cigarettes. It looks like a cigarette. The vape pen is sort of the second generation, and then there's the mod, in which we can modify the voltage, wattage, or temperature settings of the e-cigarette. And then the 4th generation pod system is typified by, or the best example of it is the Juul e-cigarette. But all of these heat nicotine flavors and solvent vehicles, being vegetable glycerin and propylene glycol, to produce aerosols. And the heating of the solvents, vegetable glycerin or VG, and propylene glycol or PG, produces aldehydes as well. So the aldehydes generated by VG are predominantly acrolein and formaldehyde, whereas propylene glycol also produces formaldehyde and acetaldehyde, and both produce reactive oxygen species. So these have great potential to have adverse effects. The acrolein itself and the particulate matter or suspended particles in the aerosol from e-cigarettes has been shown to compare fairly closely with e-cigarettes in some circumstances. So here is some data from an e-cigarette heating 100% vegetable glycerin and compared to 2 research e-cigarettes, the 1R5F and the 3R4F. The 3R4F is like a Marlboro Red. And you can see that the acrolein levels are about 2-fold in the full flavored 3R4F cigarette. But for the e-cigarette, they're still there. And a lot of users of e-cigarettes tend to puff on e-cigarettes more frequently than they might puff on a cigarette. Shown here on the right is a graph depicting or comparing cigarettes and e-cigarettes in their production of particles. So particles are tied to adverse cardiovascular effects when inhaled. And you can see that in black, the e-cigarette that they examined had comparable levels of particulate matter as the ultralight cigarette, research grade cigarette, 1R5F, and about half the PM concentration of the full flavored cigarette, the 3R4F. So this is important because several of the constituents found in these e-cigarette aerosols that I just mentioned have been shown to increase cardiac risk. And that includes acrolein, acetaldehyde, and formaldehyde, which have been estimated to contribute the vast majority of non-cancer risks in cigarette smokers. And as I mentioned, reach similar levels in e-cigarette aerosols as they do in cigarettes. Also, acute inhalation of particulate matter or suspended particles in the air, termed PM 2.5 because they're 2.5 microns in diameter or less. The inhalation of these has been shown to alter cardiac conduction and regulation of the heart through the autonomic nervous system. And exposure to PM 2.5 has also been shown to induce cardiac arrhythmia. Acute exposures to acrolein as well have been shown to alter cardiac conduction and induce imbalance of the autonomic nervous system and provoke arrhythmia in experimental settings. So I devote a lot of my time to looking at the electrocardiogram, which is probably one of the most ubiquitous clinical tools for assessing the heart's health. And it's simply a readout of the voltage generated across the body by cardiomyocytes, which have electrical activity. Abnormalities within the ECG predict adverse cardiovascular outcomes, some of which are listed here. And those abnormalities can be either changes in the shape of the ECG or changes in the rhythm of the interbeat intervals. So changes in the shape of the ECG, which are depicted here, sorry, not changes, but the shape of the ECG is depicted here within one cardiac conduction cycle on an ECG of a human. And you can perhaps appreciate the colors represent the different regions of the heart that are undergoing either depolarization or electrical discharge to induce contraction, or repolarization. So, for instance, the green area corresponds with the atria, during which depolarization across the atria is occurring on the ECG on the P wave. And then in this kind of, I guess, fleshy colored section of the ECG, that represents the ventricles and includes both depolarization on the QRS as well as repolarization that follows thereafter across the ventricles. So changes in the shape of the ECG can inform on functionality of conduction and just functionality on the heart. It's important to note that I predominantly examine mouse ECGs and previously rat ECGs. And mice and rats do have ECGs that are comparable to the human. The mouse has some important differences. So here we have a human ECG, lead 2, and in the mouse, this is a lead 2 ECG. And the one most predominant difference is that there is an inverted T wave in the mouse, whereas it's an upward deflection in the human. And this is due to a thinner fiber of fatty layer in the AV conduction system that enables depolarization from this kind of rostral end of the heart, or apical end of the ventricles. Sorry, basal end of the ventricles, and then a similar repolarization wave. So here is a magnified version of the mouse ECG, and we can examine it for changes in a number of endpoints that inform on atrial depolarization and ventricular depolarization, represented in the red here, and then also ventricular repolarization, represented in the green. So the ECG has been used to demonstrate predictivity of likelihood of death or disease. So changes in the ECG and, for instance, the PR interval or the duration of the P wave, which represent atrioventricular conduction and atrial conduction respectively, studies that have shown abnormalities in measures of those have demonstrated increased likelihood of death and disease in those individuals. So that's what's represented here, are large cohort studies showing that kind of deviations from the population average and, for instance, P wave duration, correspond with increased likelihood of atrial fibrillation and cardiovascular death, as well as risk of stroke. And similarly, abnormalities in the QT interval duration, both short on the left of the median here and long on the right of it, are connected with increased likelihood of mortality due to cardiovascular complications. In addition, the Food and Drug Administration has essentially mandated that all drugs be tested for QT prolongation. And that is because prolongation of the QT is so predictive of increased cardiovascular risk. In fact, QT prolongation is one of the leading causes of withdrawal of drugs from the market and termination of clinical trials involving new drugs. So on the other hand, using the ECG, we can look at the variation in beat intervals or heartbeats across time. And by analyzing variations in the interbeat intervals, we get indices of heart rate variability. Heart rate variability indices have been shown to decrease in value with increasing cardiovascular risk. So reductions in heart rate variability have been connected to increases in likelihood of cardiovascular complications and mortality. And increases in heart rate variability have been shown to predict reduced risk, cardiovascular risk among individuals. In addition, reductions in heart rate variability are connected to sympathetic activation and sympathetic dominance that governs fight or flight stress responses of the heart and cardiovascular system, whereas parasympathetic dominance corresponds with increases in heart rate variability. So we can use the ECG to generate heart rate variability indices, including the standard deviation of normal RR intervals. So each subsequent R wave on an ECG waveform represents a heartbeat, and the time between successive R waves is a way to measure heart rate variability. This is very sensitive to autonomic regulation, and increases in SDNN or RMSSD suggest parasympathetic regulation, whereas decreases suggest sympathetic. We also have other parameters we can generate through statistical transformations, including HF and LF, standing for high frequency and low frequency heart rate variability, that also provide some insight into autonomic regulation of the heart. And heart rate variability has been used to demonstrate likely autonomic effects of smoking and e-cigarettes. So one study showed that secondhand cigarette smoke exposure in mice increased cardiac sympathetic regulation of the heart after exposure. So you can see here reductions in RMSSD and SDNN on days following several hours of exposure to secondhand smoke in these mice. And then recently, human volunteers were shown to have reductions in HF heart rate variability after e-cigarette use, particularly if there was nicotine in the e-cigarette. And this suggested sympathetic dominance in these individuals that was corroborated by increases in catecholamines. So my interest is how do alterations in autonomic regulation and also how do constituents of e-cigarettes mediate the effects of e-cigarettes on the heart. And so I set out to compare the impacts of different e-cigarette liquids on the heart and to look at the role of the autonomic nervous system in the process. So the hypothesis of my initial research with e-cigarettes was that e-cigarettes alter cardiac physiology through aldehyde constituents and imbalance of the autonomic nervous system. So upon inhalation of aerosols with aldehydes in them, or any aerosol with irritating capacity, there is a stimulation of irritant receptors that causes neural reflexes that change the way the heart functions. And this could increase likelihood of arrhythmia, for instance, but also likelihood of other adverse outcomes. So to investigate the potential impacts of e-cigarettes on the heart, we use radio telemetry in mice and place them in exposure chambers and expose them, monitoring their ECGs real time during exposures and also thereafter by a wireless radio transmitter that sends a signal providing constant ECG waveforms. So for these exposure studies, we initially just performed acute 90-minute studies with only 3 9-minute puff sessions represented here on a timeline. So the animals were exposed for 9 minutes in the initial puff session, followed by a 9-minute washout, a second 9-minute puff session, another 9-minute washout, and a third 9-minute puff session, and a somewhat prolonged washout here. And we evaluated ECG at a 5-minute baseline immediately before the beginning of exposure, and evaluated ECG all the way through these exposures. The exposures involved either blue plus battery with a blue e-cig cartridge, and or they involved a Mystic Bridge sort of tank that we filled with e-cigarette solvents and connected to the blue plus battery. We also compared impacts on mice exposed to mainstream cigarette smoke, or MCS, using combustible cigarettes. And those exposures were slightly different. They only involved 2 9-minute puff sessions, just because the use of cigarettes usually is less frequent to attain the same level of nicotine. And we also looked at the impacts of acrolein gas exposure in a separate study involving 2 9-minute continuous exposures. So this is a summary of all the comparisons we made, all the different exposures we conducted, and included in these different exposures were 100% propylene glycol, 100% vegetable glycerin, a 50-50 sort of even distribution of PG and VG in a single solution, because these tend to be mixed together in e-cigarette e-liquids. And then for the comparison to the blue e-liquids, we compared classic tobacco to magnificent menthol. Each of these have 2.4% nicotine at a 70-30 PG-VG ratio, so predominantly PG within them. And then here's a summary of the mainstream cigarette smoke cigarettes that we used. The 3R4F is much higher, basically 10-fold higher in tar and in nicotine relative to this ultralight 1R5F. And then the aldehyde acro, which was at 3 ppm. So the first most obvious thing we observed was that there was a dramatic reduction in heart rate in the initial minutes of exposure. So here is an average of 4 animals immediately prior to exposure to PG-VG e-cig aerosols. And you can see their heart rate's about 700 beats per minute. But within the first minute, the heart rate is dropping rapidly. And within about probably 4 minutes, the heart rate is bottom out to about 320 beats per minute, and rebounds only after the cessation of the puff session. So this is a 9-minute puff session, and these peaks right here represent each individual puff in terms of the total suspended particulate, which we measured using a densitometer or densitometer. And then you can see during the washout period, heart rate rebounds rapidly, and then in the subsequent puff session, it drops again. So that's the impact of PGVG. That was really dramatic, but we also compared the different aerosols, and shown here are 1 minute mean values in terms of change from the baseline value of heart rate. And you can see that 100% PG had a slighter effect than 50-50 PGVG. VG had a fairly comparable effect to PGVG. And then if you look at classic tobacco and magnificent menthol, the impacts are extremely subtle. They're reduced. And it wasn't clear from this data whether maybe the nicotine in these e-liquids was the cause of this diminished impact. But nonetheless, another notable aspect of this data was that late after the exposure, we saw elevations in heart rate when compared to the air control exposure in the open circles shown here. So there seems to be opposing effects on heart rate. And when we analyzed the average response across all 3 of the 9-minute puff sessions, this was, the average effect on heart rate of these different exposures. Then looking at the sort of washout periods, which I'm calling early post, occurring within the first 9 minutes after each puff session, we saw that there was a transition from this reduction in heart rate to, in some cases, increases in heart rate for the nicotine-containing e-liquids and PG, and in other cases, a persistence of the reduction in heart rate. And then late after exposure, from approximately 10 to 30 minutes after the final puff session, we saw increases in heart rate for almost all exposures, suggesting a switch from this reduction in heart rate during the puff sessions to an increase in heart rate afterwards. So we wanted to determine what the impact on regulation by the autonomic nervous system of the heart was. And our hypothesis was that e-cigarettes would induce proarrhythmic alterations in autonomic balance similar to mainstream cigarette smoke and acrolein. And we saw that, for instance, looking at heart rate variability measured by SDNN, the change in heart rate variability from the baseline values was much higher with the nicotine-free solvents than it was with the nicotine-containing e-liquids in general. But the effects were generally comparable to mainstream cigarette smoke and to acrolein alone. And all of these effects during exposure were indicative of irritant reflexes and parasympathetic modulation of the heart. And then after exposure, we saw reductions, for the most part, in heart rate variability, both in that washout period as well as the late post-exposure period at 10 to 30 minutes after the final exposure session. And this suggested sympathetic dominance after exposure for virtually all of the e-cigarettes and cigarette or acrolein, with one exception, the full flavored cigarette, which I don't have an explanation for, but nonetheless, the consistency is what I'd like to focus on here, that most of the exposures caused a fight or flight sympathetic response late after. So what were the impacts on cardiac conduction? We looked at a number of intervals and amplitudes along the ECG, and this was to address the hypothesis that e-cigarette aerosols would induce changes in the heart that promote arrhythmia. And we saw that during exposure, e-cigarettes altered atrial and atrioventricular conduction. First, looking at atrial conduction, or P duration, we saw generally e-cigarettes increased or prolonged atrial conduction times, with one exception, that menthol, magnificent menthol, accelerated atrial conduction time. And also the ultralight cigarette seemed to accelerate atrial conduction time as well. So P duration is represented here. And then we also looked at PR interval, which indicates atrioventricular conduction time. And typically, these would correspond with each other to some extent, but we found that magnificent menthol uniquely prolonged atrioventricular conduction time despite accelerating atrial conduction time. So this was somewhat, I guess, confusing. But if you look across the other exposures, for instance, we see with the ultralight cigarette that there is a reduction in PR interval concurrent with the acceleration of P wave duration. So that's more consistent. The opposing effects on atrial conduction and atrioventricular conduction could produce arrhythmia because they're working in opposition to each other, and this could cause a dyssynchrony in conduction patterns and rhythms. So we also looked at the QT interval, which I mentioned earlier. So this is a measure of ventricular repolarization time, and we saw the e-cigarette solvents dramatically prolonged the QT interval, especially PGVG. And PGVG itself did so more than even the full-flavored cigarette. But the e-cigarette solvents and the mainstream cigarette smoke and acrolein all prolong QT interval during exposure. So shown here is just a representation, one animal before in blue, before PGVG exposure, in terms of 1 minute average ECG waveforms. So each one of these lines is a 1 minute average, and in this case in blue, preceding exposure, and then following the end of the first puff session, here we have kind of the post-exposure effect on the ECG, and you can see the QT interval from here to there before exposure, and then from here to there after exposure was prolonged. You can also see this PR prolongation here. So early after exposure, the QT prolongation persisted with the e-cigarette solvents, and it was also evident to some extent with the nicotine-containing blue e-liquids, flavored e-liquids. And then late after exposure, there was still slight but significant prolongation of QT interval with the e-cigarette solvents. So some of this data, especially the PGVG and the VG data, especially in the early post-exposure here, as well as the acrolein here, suggests that acrolein-rich aerosols could promote arrhythmia by slowing ventricular repolarization. So acrolein slowed repolarization. We know mainstream cigarette smoke has acrolein in it. And then VG disproportionately produces acrolein. So that might explain why there appears to be a disproportionate impact on ventricular repolarization by these VG-containing e-liquids. So here's sort of a summary. It's, I realize, a little overwhelming. You're probably, even without the summary, overwhelmed. But here is a summary of all the effects, I'll back up here, that we saw with the e-cigarettes, separated by mid-exposure, this early post-exposure, and the late post-exposure phase impacts on the ECG shape. And you can see that there were some unifying effects. At late post-exposure, P duration was decreased, and PR interval was decreased as well for all of the exposures. And then, there's some oddities here. For instance, menthol shortened P duration, but prolonged PR interval, as did PGVG, but PGVG didn't impact P duration like that. In terms of repolarization here, across the board, we saw QT prolongation. So that was particularly intriguing and has clinical implications, but we'll look at that further in a minute. Okay, so we also looked at heart rate and heart rate variability, and pretty much universally, reductions in heart rate during exposure with increases in heart rate variability. And then after exposure, increases in heart rate along with reductions in heart rate variability, suggesting this phasic response of parasympathetic kind of irritant reflexes during exposure, followed by sympathetic fight or flight reflexes after exposure. Okay, but what about actual arrhythmias? Well, we looked at those, and I'll get into that in a second. But I just want to explain some of the clinical significance of one type of arrhythmia. It's called a ventricular premature beat. Some people call it premature ventricular contraction. It has maybe a few other names, ventricular tachyarrhythmias. But a recent publication demonstrated that, and we already know that these occur pretty frequently in people, especially with age. They're not totally unusual, and they're not generally considered extremely alarming to observe clinically. But this study was a large population study that looked at ventricular premature beat frequency, how often they occurred in a given day, and whether this was predictive of mortality. And the authors found that as few as just 1/2 of a ventricular premature beat per hour, so that's basically 1 every 2 hours, was enough to predict an increase in likelihood of death due to cardiac complications. So that's the differentiation here is broken down into number per day. So this is if you had less than 12 per day, or less than 1 every 2 hours, then here's your risk for cardiac mortality. If it's greater than that number, then here's your risk, which is basically twofold. And similarly, heart failure actually the risk was tripled. Sudden cardiac death, the risk was increased 6-fold. And so the point I'm trying to make here is that ventricular premature beats are clinically significant arrhythmias. And we saw with e-cigarette exposures in these mice that propylene glycol alone, and also magnificent menthol, those exposures increased significantly the number of ventricular premature beats to a mean frequency, I believe, of about 2 to 3 ventricular premature beats per hour. Now, of course, we're comparing mice to humans. Mice have more heartbeats per hour than a human does. So that kind of cutoff point that I just explained for humans is probably different than it is for mice. But nonetheless, we did see increased frequency of these arrhythmias, and this could be a clinically relevant observation. So interestingly, also, we saw that the impacts of e-cigarettes on the QT interval corresponded with the occurrence of these ventricular premature beats. So changes in repolarization of the ventricles may be connected to the appearance of these arrhythmias. And this was nicotine dependent. So if you look at the blue values shown here, we found that for the nicotine-free, like no nicotine in the e-cigs, just the solvents, there was a positive correlation between QT interval and ventricular premature beats. Whereas for the nicotine-containing e-liquids or cigarettes, we saw an inverse association between QT interval and ventricular premature beats, such that a shortening of QT interval corresponded with an increase in ventricular premature beats. And this is kind of the breakdown. Basically, for a 1 millisecond increase in QT interval, that predicted a 10% increase in ventricular premature beats. For nicotine-free exposures, but with nicotine-containing aerosols, a 1 millisecond reduction in the QT interval predicted a 25% increase in ventricular premature beat. Okay, so that hopefully was an interesting clinical connection, maybe, and also it introduced the concept that nicotine could be modifying the effects of the cardiac effects of e-cigarettes. But I want to return back to the autonomic nervous system and its potential role in the effects of e-cigarettes. So it's already well appreciated that the autonomic nervous system mediates the cardiotoxicity of inhaled pollutants. And that's been demonstrated by a number of studies. I cite some of mine here, but there are far, far more than that have shown that exposures to high concentrations of aerosols, pollutant aerosols, for instance, causes a biphasic autonomic imbalance, where initially there's a parasympathetic dominance followed by a sympathetic dominance. And in addition, inhaled pollutants have been shown to increase arrhythmia risk through imbalance of the autonomic nervous system and through irritant pulmonary reflexes, as kind of depicted here. And so we set out to test the role of the parasympathetic branch in the impacts of solvent-only e-cig exposures. So exposing animals to PGVG with a pre-treatment to either saline or atropine, a parasympathetic blocker that inhibits the muscarinic receptors, we found that just these acute exposures that I showed earlier, basically replicating those exposures, we saw that the impacts of PGVG on measures of ventricular repolarization were diminished significantly by blockade of parasympathetic regulation of the heart, at least during exposure. And this indicates that the parasympathetic branch mediates some of the effects of e-cig aerosols on cardiac conduction. And it may underlie possibly the arrhythmogenic impacts of e-cigarettes. In addition, we were interested in the irritant reflexes, and one of the prevailing molecular mediators of pulmonary irritant reflexes in cardiac effects to irritants. So we wanted to look at the role of the TRPA1 irritant channel, which resides on C fiber neurons that mediate the irritant stimulation of the lungs and autonomic reflexes. So we wanted to test the role of the TRPA1 irritant channel in the effects of e-cigarettes on the heart. And the reason why we wanted to do this was because there's already precedent observed with exposure to aldehydes and air pollutants, demonstrating that that the TRPA1 is responsible for the arrhythmogenic effects of these exposures. And it may be responsible through a sympathetic receptor, the beta adrenergic receptor, which is one of the main autonomic receptors on the heart. But also, the TRPA1 has been shown to mediate parasympathetic responses to acrolein in other studies. So we acquired a mouse that is a TRPA1 knockout. It's a global knockout. It does not express the TRPA1 irritant channel, and we exposed it to PGVG aerosol, nicotine-free, and looked at the impacts on heart rate. We found that here in green, when exposing these knockouts to PGVG, we did not see the bradycardia that we otherwise observed in wild-type animals. So here in the dark blue, you see a dramatic reduction in heart rate in the wild-type animals and a rapid recovery thereafter. The open circles represent the air-exposed wild-type animals in blue, and open circle green is air-exposed TRPA1 knockouts. So we also tested the potential role of the beta adrenergic receptor in these responses by administering a beta blocker, propranolol, and found that, as indicated in the kind of light squares, that beta blockade prevented the rebound in heart rate after exposure in wild-type animals, and its impacts were not as clear in the knockouts, simply because we didn't have many impacts to begin with in the knockouts. We also looked at heart rate variability, and it was clear that the impacts of PGVG aerosols were virtually abolished in the knockouts. And beta blockers attenuated the impacts, at least at the initial exposures, attenuated the impacts on heart rate variability. So it's also noteworthy that late after the exposures, we had elevations in heart rate and reductions in heart rate variability in the wild type animals. And to some extent, these were diminished in the knockouts, but not completely. So from this data, it was evident that the TRPA1 at least partly mediates the changes in heart rate and autonomic modulation. And I've just got to summarize it here with respect to the different exposure phases as previously. But on the left, we have the wild types normalized by the air exposure kind of time-matched values on the left. And the values are changed from baseline. So we did separate air exposures in both the wild types and the knockouts. And then, you know, here's the relative bradycardia in the wild types and the absence of it in the knockouts. Sorry. Early after exposure, we see a relative tachycardia in the wild types and not so in the knockouts. Late after exposure, this tachycardia becomes more prominent in the wild types and is virtually non-existent in the knockouts. And then with beta blocker, you can see that the reduction in heart rate is slightly diminished. This might have been due to a lower resting heart rate at baseline in the first place. But you also see inhibition of the increases in heart rate, at least early after exposure in the wild types. But this benefit is lost by the time that we get to the late post-exposure period for reasons unclear. So interestingly, late after exposure, we did see that TRPA1 knockout only partly attenuated the reduction in heart rate variability induced by e-cig aerosols. And beta blockade also attenuated impacts on heart rate variability, but only partly as well. And when we combine beta blocker with TRPA1 knockout, we see a combined impact suggesting that the TRPA1 irritant channel and the beta adrenergic receptors both kind of jointly but independently mediate the reductions in heart rate variability or sympathetic regulation of the heart after e-cigarette exposure. We also looked at arrhythmias and saw that in the wild type animals, parasympathetic blockade did not seem to inhibit the arrhythmia induction. So here's a saline pre-injected animals values, mean across, I believe, an N of 4. And then here are the number of ventricular premature beats with atropine pre-injection at either at one of 2 doses. Beta blockade did not seem to prevent the effects either. And notably, beta blocker was administered through drinking water as opposed to injection. So we should kind of compare visually between the regular drinking water animals without any injection and this propranolol value. But when we looked at the TRPA1 knockout, which is in gray, so in black is the same data that I was just showing you here over on the left. But in gray, here we have the knockouts included. You can see a potential dose-dependent reduction in ventricular premature beats with atropine. So this suggests possible complementary roles of parasympathetic regulation and TRPA1 in e-cigarette-induced arrhythmia. So from this study, it was evident that e-cigarettes disturb autonomic regulation of the heart and conduction of the heart. Similar to cigarettes and acrolein, they have phasic effects on heart rate and heart rate variability. And those effects are mediated by the TRPA1 irritant channel and autonomic receptors. It's also clear that e-cigarettes induce arrhythmogenic changes in the heart's conduction. I'm not going to go through each one of these, but you can review if you're a little lost here. But the implications are multiple, and just some of them here are that if these effects are confirmed in humans, government agencies may choose to regulate the chemical constituents within e-cigarettes that promote arrhythmia, and they could require pre-market testing of e-cigarette products for potential impacts on electrophysiology. Government agencies and public health officials could also issue advisories for susceptible populations considering using e-cigarettes, such as those with underlying long QT syndrome or survivors of myocardial infarction. So now I'm going to switch over to Juul, which is the latest and greatest, I suppose, e-cigarette du jour. And right here I've titled it "Fooling the Next Generation of Nicotine Addicts," but it's also fueling the next generation of nicotine addicts. And this is especially evident when we look at the past kind of 5 years or so of e-cigarette use. So if you look at this chart here, the colors or the successive bars represent successive years and the prevalence of e-cigarette use among high schoolers in the United States. And you can see from 2017 to 2018, we have this rapid increase from about 12% to 21% in use prevalence for e-cigarettes. And that was fueled primarily by Juul's introduction to the market, which is extremely popular among youth. Juul is also really high in nicotine compared to the other e-cigarettes before it. And the nicotine, it comes in a different form, nicotine benzoate, making it more palatable to have these high levels of nicotine. As a consequence, adolescents recently tested who were using Juul had higher urinary cotinine than historic tests in adolescent smokers, and considerably higher urinary cotinine. So this is cause for alarm. It may be that teenagers who are choosing to use tobacco products now are getting much higher doses of nicotine, potentially much more addicted, and nicotine itself could have adverse impacts itself. So in addition, there are flavors in these e-cigarettes, and they may also contain unique toxins within them. So to illustrate that, shown here are, I believe, let's see, 6 different, or 8, 1, 2, 3, 4, 5, 6, 7, 8 different flavors of Juul, and kind of a duo of tests or duplicate of tests of the chemical constituents within each of these different flavors. Most of these have been banned essentially in the United States, such that it's just menthol and tobacco now for Juul. But what you can see here is that there are a number of chemical constituents with potential irritant capacity or injurious effects that are heterogeneously present in some e-liquids versus others, and at varying concentrations. So the different flavors could have different effects on the heart. So we set out to test, somewhat similar to our prior studies, but involving a 6-hour exposure instead of just that 1 hour 3-puff session exposure, the impacts of Juul e-liquids on mouse cardiac conduction. And the 6-hour exposure involved 20 9-minute puff sessions. We exposed animals to an air control and compared their responses later to a PGVG exposure at the 30-70 ratio found in the Juul e-liquids. We then also compared the impacts of 3 different Juul flavors, e-liquid flavors. And so we saw that across the 6 hours, the mean impact on heart rate with the e-cigarette solvents involved a reduction in heart rate with the PGVG when normalized by the air value, and an increase in heart rate for the nicotine-containing e-liquids when normalized by air value. And actually, these impacts were variable with significant differences between the groups, the different Joule flavor groups. Across the 6-hour period by hour, we can see that there is a time-dependent effect, especially a PGVG, where the heart rate reduction becomes all the more dramatic by the 6th hour of exposure. It's not nearly as clear for the nicotine-containing e-liquids. And then heart rate variability followed a similar pattern for the PGVG exposure. So from this data, it was evident that the presence of nicotine altered the impacts of e-cigarettes on heart rate and heart rate variability. We also looked at ventricular premature beats and saw that the e-cigarette solvents without nicotine caused the greatest increase in ventricular arrhythmias, and mango was sort of the next greatest impactor on arrhythmia frequency, but all of these exposures significantly increased ventricular arrhythmias. So from this, it was evident that nicotine and flavors modified the arrhythmogenic effects of e-cigarettes. We also looked at, across the 6-hour period, the impacts on ventricular premature beats, and you can see, like heart rate variability, PGVG increases ventricular premature beats across that 6-hour period, and the highest values were observed during the highest values of heart rate variability. So we analyzed the corresponding difference in heart rate variability measure of SDNN from baseline and frequency of ventricular premature beats at a given period for each animal, and found that there was a significant positive correlation between heart rate variability and ventricular premature beats. So increasing heart rate variability was predictive of an increase in ventricular premature beats. It was also evident from this that nicotine could acutely attenuate the arrhythmogenic effects of e-cigarettes, and likely through the sympathomimetic effects of nicotine itself. Okay, so I'm going to segue quickly into kind of a summary of just a few of the translational observations we've made comparing humans to mice. And these aren't direct comparisons, but I just wanted to illustrate how our observations in mice may pertain to the effects in humans. So just a reminder, the ECG of the human and the mouse compare, but not perfectly. And a former PhD student of mine, now at the Mayo Clinic, he's a cardiologist now, he and I recently published a study showing that in smokers, atrioventricular conduction, as measured by the PR interval and PR segment, was shortened, or the atrioventricular conduction was accelerated, corresponding with increases in nicotine or cotinine. And the shortening of the PR segment, you can see, is correlating with increasing cotinine. That shortening, what we determined statistically, was likely mediated by increases in dopamine. So here's the PR segment and its correlation with cotinine. And then here is also, in a follow-up study in a large population in Haines in the United States, we validated these observations of cotinine's correlation with shortened PR segment. And so this suggests that smoking through nicotine accelerates atrioventricular conduction, and likely through increases in dopamine. So in mice, meanwhile, in that 6-hour kind of Joule e-liquid exposure study, we saw that the nicotine-containing Joule e-liquids significantly reduced PR segment when compared to the E6 solvents, and at least 2 of the Joule e-liquids with nicotine in them significantly reduced PR segment or accelerated AV conduction. But also, the Joule e-liquids increase urine dopamine, these Joule e-liquids with nicotine in them, whereas PGVG alone without nicotine used in a Joule device did not significantly increase dopamine. And we also looked at the effects of a mod operated at varying temperatures with PGVG alone and did not see a consistent increase in dopamine with exposures. So from this, it was evident that nicotine-containing e-liquids accelerate atrioventricular conduction and increase urinary dopamine, consistent with our observations in smokers and dopamine and cotinine levels along with PR segment. So in conclusion, from our studies, it is apparent that e-cigarettes acutely change the conduction of the heart and the regulation of the heart by the autonomic nervous system, and they promote arrhythmia. The autonomic nervous system and TRPA1 irritant receptors appear to be responsible for several of the effects on the heart, and the arrhythmogenic effects of e-cigarettes also appear to be prevented by nicotine and modified by the presence of flavors. For instance, we found that menthol, at least the blue menthol e-liquid, induced a significant increase in arrhythmias. Also from our studies, it's apparent that smoking accelerates atrioventricular conduction and increases dopamine in humans through nicotine, and we've seen similar impacts in mice. So overall, from these observations, it's apparent that chemical constituents, for instance, nicotine and flavors, can affect and also underlie the actual impacts of e-cigarettes on the heart. So targeted regulation by government agencies of e-cigarette chemicals, and also the types of e-cigarettes, their operational settings, user settings, those may be targeted by government agencies in order to reduce harm. So with that, I thank you for your attention, and I'd just like to acknowledge many of my colleagues, my lab members shown here, my funding, including funding that supported Claudia Arab and Renata Salatini to visit through CAPES for a 1-year sandwich experience in my lab. And if you have any questions, please feel free to email me. My Twitter handle is shown here. You can hit me up on there, I guess. So thanks so much for your attention, and I appreciate this opportunity to introduce to you some of the important effects of the cigarette on the heart. I can't hear anything. I don't know if you're muted or what's going on with my audio. One second. Sorry. I could hear my talk, but I can't hear anybody speak. I can't hear Leticia speaking. Don't hear anything. I see Flavia and Leticia, and I can't hear Leticia. I'm sorry. I can see in the chat. Yes.
So can you all hear me? Can you nod if you can hear me speaking? Okay. Well, that's easier. Yeah, so I do intend to continue researching air pollution. And I actually do intend to compare the effects of electronic cigarettes to those of air pollution when it comes to electrical remodeling of the heart, so altered expression or phosphorylation of cardiac ion channels and other proteins within the heart that regulate calcium handling and are also targets of the autonomic nervous system. So my research is mostly a two-pronged endeavor in that, you know, I don't want to abandon my interest in air pollution. I certainly want to continue after them. But presently my funding is entirely for e-cigarettes. So that's kind of where I have to focus my energy. But comparing the two is very much an interest of mine. Just outright, the comparability is it's already there for the making to some extent, but the air pollution exposure levels in the ambient air, those air pollutants usually are at much lower concentrations than in e-cigarette aerosols, unless we're talking about maybe secondhand e-cigarette aerosols. So the particles, the formaldehyde and acrolein, all are going to be at much lower levels in the ambient air, usually, than with respect to e-cigarettes. So you can see actually sometimes opposing responses that seem totally contradictory, like they completely disagree with each other. But the kind of the physiological mechanisms actually explain why they would be in opposition to each other. So that's very much of interest to me as well. Thank you for a great question. Yeah. Well, I think the true risks of electronic cigarettes themselves are just being discovered, when it comes to users, and even for users, because I want to address that first and foremost, since that's probably the greatest risk. In terms of the clinical data, the blood pressure impacts of e-cigarette use, the impacts on heart rate and also the impacts on like flow mediated dilation or vascular function are diminished relative to smoking. That doesn't mean e-cigarettes are safe, but they may be less harmful, possibly, than cigarette smoking. So they could be a reasonable alternative to smoking if a smoker wants to transition to e-cigarettes completely. That could be to their benefit. And there's some evidence that indicates that they could be helpful for smoking cessation. And ideally, the user would then quit puffing on an e-cigarette after that. When it comes to secondhand exposure, very little is known. And generally speaking, I think that it's also going to be dramatically less harmful than secondhand smoke. But there's all sorts of possibilities. The nicotine in the aerosol, the flavors, especially the flavors, have a there's so little understood about what the impacts of inhaling the different kinds of flavors, and there's like over 7,000 different e-liquid formulas out there, with all kinds of different flavorant chemicals that compose individual flavors. Like menthol, it's not just menthol, there's all kinds of flavorant chemicals in there. So it's yet to be known, the risks for secondhand smoke, and it's something I'd like to investigate further. The NIH is funding research into the secondhand exposure effects. Presumably they'd be less because they're at a lower concentration, but we don't know that for sure. And especially for people who have underlying health problems, asthma, a genetic predisposition to arrhythmia, stuff like that. So overall, the risk, I don't know. There could be an increased risk for arrhythmia. Why did I only test TRPA1? That's a good question, because there are definitely other relevant irritant channels, other TRP channels that reside on sensory C fibers, C fiber neurons that trigger reflex, cardiac reflexes that can promote arrhythmia. So there's TRPV1, there's TRPC, M8, which is the menthol TRP channel. So it's stimulated by menthol. And TRPA1 is, it's one of the hottest TRP channels out there because it's stimulated by a number of things that are already understood to be toxic. Acrolein being high among those, formaldehyde as well. And if you look at the non-cancer risk of smoking, roughly 90% of it is estimated to result from acrolein and formaldehyde and acetaldehyde. All three of those stimulate the TRPA1 irritant channel. And so because they're potent stimulators of the TRPA1 irritant channel, that and they're found in e-cigarettes, it was kind of the ideal candidate irritant channel to look at in the mouse. So that's why we only tested TRPA1. Could other TRP channels be involved? Absolutely, especially when it comes to menthol and TRPM8. But certainly, it's a good question. Yeah. Oh, sorry. And the other channels involved with ROS. Sorry, could you? Well, to be honest, my understanding of the diversity of channels that are stimulated by ROS is limited. Mostly stemming from the fact that I'm a gross physiologist by trade, I guess. And, I think there are a multitude of them that are stimulated by a reactive oxygen species. TRPA1 is one among many. So yeah, it's a great point. I think that generally, at some point when you start conducting mechanistic studies, you kind of have to settle for reality, which is that there's no way to kind of look at all of them. There's certainly reason to look at others, but if you have any suggestions about particular receptors and channels really that should be examined, especially when it comes to reflexes, I'm all ears. I mean, there are cardiac ion channels that are affected by reactive oxygen species as well, and it's possible that cardiac oxidative stress is underlying some of the arrhythmic effects. But we did want to hone in on sort of this reflex control of the heart because the effects seem so immediate. So that maybe reduces some of the irritant channels or the channels down to those that reside within the airways and lungs. But if you have any suggestions, I would love to hear about things, channels that you think would be wise to look at. No, your microphone didn't break. For some reason, the audio it shut down on me. I could hear myself speak, which is useless, but when the talk ended, I couldn't get any audio from you guys. Okay, so I see the next question, and I'm happy to entertain anything else that was said after this, but will I analyze brain nuclei? So I'm absolutely interested in looking at different regions of the brain that could be impacted, and actually, one of the major reasons why I'd love to look at different regions is because central oxidative stress is a potential underlying cause of the effects as well. But also there are irritant receptors that are located in the brain that are stimulated by the C fibers after the TRPA1 or other trips are stimulated. For instance, the P2X receptor. But yeah, I mean, there's different regions of the brain. It requires a specialization to know how to isolate them that I lack, and currently nobody in my lab has. But if you have the ability, by all means, please reach out to me. Maybe we can arrange something. I'm interested in the nucleus tractus solitarius, the paraventricular nucleus of the hypothalamus, the rostroventrolateral medulla. I mean, there's a bunch of places I'd love to look, but I mean, in a mouse especially, I don't know how to isolate them. And I have folks that might be able to help me someday, but you know, having somebody in your own lab who knows how to do that would make a huge difference. I hear somebody now. Giovanna? Whoa.
Hey, Alex. Are you
Hi.
hearing me?
Yes, you're the only one.
Okay. Thank you so much for your time and for the class. It was really amazing,
Thank
and
you.
for answering our doubts.
Yeah, my pleasure.
We're from Brazil, right? And I'm sorry for Leticia because she had some problems with her audio.
Well, she's not the only one, right? I mean, I think others were asking questions. I couldn't hear them, so it may not be on your end. It might be on mine. I don't know. But no problem at all. And I really appreciate your all's attention. You know, I've been fortunate enough to work with a lot of Sao Paulans, Sao Paulistas. Is it Sao Paulans, right?
Yeah, Paulistas.
Oh, Paulistas. Okay, all right, okay. You know, over the years, and I'm honored that Talita invited me. And you know, I've been really impressed with the work ethic and discipline that all of the Sao Paulistas that I've worked with in the past have shown. So it's my honor to be able to give a little talk to you guys and to actually see you ask good questions too. I appreciate that a lot.
Oh my God, thank you so much. Really, it was our honor to receive you here. And we hope for some more classes, right?
Yeah. I mean, hopefully, my heart goes out to you guys. I know you're getting hit hard by COVID-19. At least that's what they say. And you know, the United States is maybe not the best example for pointing fingers at somebody else, but I hear Brazil's pretty bad right now. So stay safe, and you know, I also have hopes that the CAPES funding will sort of improve over time, and you guys will have more opportunities to come to the United States.
Yes, but you're right. Here, the situation is not of the best, but we will continue to be safe here. Thank you so much.
Thank you. It was a pleasure meeting you. And Leticia, I'm sorry I didn't get to hear your voice, but
Yeah.
I can always get another talk.
Yeah, you're seeing her, right?
Yes.
Okay, so thanks, Alex. We will
Thanks so much.
now finish the event.
Okay, great. Feel free to email me. I didn't provide my email address in the talk, but if anybody needs it, just send Talita or anybody else an email or look me up.
Thank you so much. Good
Thank you.
night.
Good night.
Bye-bye.
Bye.